Okay, I want to walk you through this chapter a little bit. The cell was discovered by Robert Hooke in 1665, and he was looking at cork. And he named it the cell because when he looked at this, it looked like monastery cells all in a row. And so that's why he named it. Also a glimpse of the microscope, pretty awesome. The cell theory, everything's composed of cells. They're basic units of structure and function, and they only come from pre-existing cells. They don't spontaneously rise like they thought long ago. Size-wise, we can see a lot of things with the unaided eye. This is just looking at that comparison. And down to a frog egg, it's pushing it. Better seen probably with a light microscope, a compound microscope. And you'll see that these overlap all the way down to the electron microscope, that you're going to see viruses and proteins and smaller organelles, so kind of cool stuff. So let's look at some pictures of those. Here's using a light microscope. There's a pine needle, beautiful. Human cheek cells, this sloughs off inside your mouth constantly, and there's the nucleus. There's a white blood cell. Then the transmission electron micrograph, TEM. We're starting to look at things smaller. Here's herpes simplex virans, so those are the viruses. Here's cell organelles, and here are mitochondria. Then the scanning electron micrograph is this new technique, or kind of new, that they actually coat it with gold, and then they get a three-dimensional look like this. So blood cells, looks like some white blood cells are there with red blood cells, and cholera. So pretty darn beautiful stuff. So what determines the size of a cell? Well, surface area to volume ratio. It has to have enough surface area to that volume so that it can absorb everything it needs and get everything out. This big square, this big block, not enough surface area for this much volume. So how does our body kind of make it better? Like the cells in our intestine, we have these tiny finger-like projections that come out of them, just making more surface area. We'll see that coming up again. So size does matter. I have a bad sense of humor. Okay, so we're going to compare prokaryotes and eukaryotes. Bacteria are what you're familiar with, and archaea are these extremophiles that we thought were bacteria, but then we saw their DNA and they're not, but they look just like bacteria to us, but live in these crazy extreme places. Everything else are the eukaryotes. Animals, plants, fungi, protists. So let's talk about those prokaryotes. Prokaryotic means before kernel, and they're talking about before nucleus. So every time you get a question on a test that if they give you an example and it does not have a membrane-bound nucleus, it's going to be one of these guys. It's either going to be a bacteria or an archaea. These guys are small, very successful, all over the place. We think they were the first cells on Earth, actually. Prokaryotic cell is very, very simple structure. We'll spend a whole lot more time looking at the structure of the eukaryotes. So generally what we're familiar with are the bacteria. We identify them by shape. I put this in just simply because it was interesting. Here's anthrax, strep throat, which you're all familiar with, I'm sure, rat bite fever, and Lyme disease are these various shapes of bacteria that we can identify. Okay, what do bacteria have? They have a cell membrane. Who else has it? Eukaryotic cells. Everything has a cell membrane. At the end of this, I'll be talking about cell membranes in detail and how they work. This is a cell membrane right here. So remember that everything's got it. It's a phospholipid bilayer with embedded proteins. We'll talk more about that soon. So the cell wall. The cell wall of bacteria maintains this shape. And an interesting thing here, it's made up of peptidoglycan and in different quantities if it's a different kind of bacteria, gram-positive, gram-negative. You might have heard that before. So penicillin doesn't let this work as well, and so these guys die. Doesn't impact gram-negative. And so that's why they'll check to see what you have if you have a culture done, if you have an infection. Cytoplasm. It's a semi-fluid inside here. It's got inorganic, organic molecules. It's a lovely mix of rough resources to use. And they have a nucleoid region. It's called nucleoid, sorry, I'm stuttering. It's not a true nucleus. It's a location where the DNA strand is located, but it is not a membrane-bound nucleus. And so that's what makes these different. However, they have ribosomes. These are where proteins are made, And so both groups have them. Eukaryotes need them too, but these are smaller and kind of free floating. Bacteria have appendages. You see the flagellum or flagella, and that helps them move. They hook onto surfaces through pili, sometimes fimbriae, and glycocalyx, which is the sticky outer coating. I'll have another slide on that. They can do this crazy thing about transferring genetic material straight across through this sex pili. It's not sexual reproduction at all, and they can do it between different species, and it's called horizontal gene transfer, and it's actually one of the reasons bacteria can mutate so quickly. So here's more on that, or can evolve so quickly. So let's say this kind of bacteria is resistant to an antibiotic. It can, by some mutation, crazy, random thing, transfer that into another one that's a different species of bacteria, but now it's resistant to that same antibiotic. So it's kind of an amazing thing. Fimbriae, or Fimbria 1, Fimbriae mini, are these little stick-like, or little finger-like projections all over the outside, and disease-causing things often use those to attach and hold on to the host. E. coli lives in our stomach, in our guts, I mean, not in our stomach, in our intestines, and they hook on by the same thing. Streptomycin, how does it work? It's kind of cool. It combines with the bacteria's ribosomes, so they don't work. Guess what? They can't make any more proteins. You're going to find out that they need to have a lot of proteins to function, and so it can't continue functioning at all. Let's say you're looking at something, and like the test question says, they look like eukaryotes. They got a bunch of stuff in there, but they have no membrane-bound nucleus. If there's not a nucleus, it's got to be a prokaryotic cell. So your choices are a bacterium or archaea. Archaea, those extreme things that live in really hot, really cold, really salty, really acidic, that kind of stuff. And here's the extremophiles, the kind of places that they might look. We just discovered them not long ago by looking at the bacteria, looking at the DNA, and realizing they were not bacteria. All right, eukaryotes. Those are the animals, plants, protists, and fungi. So the size can be anywhere between 10 to 100 micron, excuse me, I'm unable to speak today, so yes. So here we are. We've got an egg, we've got a red blood cell, we got a neuron there. So let's talk about some of this stuff. DNA is enclosed in a double membrane nucleus. That's what makes them different. That's what makes them special and more evolved. We keep the DNA separate from the other activities going on in the cytoplasm that could damage the DNA. We don't want to damage the DNA. So these pores that you'll see in the nuclear envelope here of this is the nucleus, allows the passage of certain molecules to come out, like we need messenger RNA to come out and ribosomes to come out. We'll find out more about what they're doing later, but they're building proteins. So let's do an analogy and kind of work through this as an analogy. So let's say the cell is a factory. The nucleus is information central. It's where all the genetic information and everything is. And in your factory, it'll be where your plans are for what you build, your future projections, or whatever, and all the equipment information that you need to build it. In there is going to be chromosomes. That's where DNA, genes, and our actual genetic coding are. In your factory, that would be the plans or blueprints for whatever you're building. In your body, these really code for the proteins that make your eyes the color they are, for example. We'll do lots with genetics coming up. So chromosomes are really condensed chromatin. Chromatin is the form it's usually in the nucleus, and just before it starts to divide, it condenses until it looks like this. And if you look through a microscope, these are cells, these few here are in stages of mitosis, so they're dividing, and so they're starting to condense. So you can see those chromosomes. All right, the next thing I want to talk about in the nucleus is the nucleolus, and that's what produces ribosomal RNA. That goes out to the factory floor. It takes plans with it, with the ribosomes, and so that's where that's built. Okay, the next step in our cell analogy is the endomembrane system, and that incorporates all of these things here that we'll be talking about in a second. And if you think about it, for your factory, it's the entire manufacturing and shipping for that factory. It makes whatever it's making and it ships it out to wherever it needs to go. It is going to include all of these things: the rough endoplasmic reticulum, and the ribosomes where proteins are made, and then they get in these transport vessels and move them over the Golgi apparatus, where they might modify them a little bit, add something to it, and then package it and ship it out wherever it's going to go, or get digested, whatever. All right, so you got kind of an idea on that. So the endomembrane system is all of that stuff together. So let's break it down. Here's the endoplasmic reticulum. That includes the rough ER, there's the E and R, and the smooth ER. Those are these guys right here. This rough one is called rough because it has those little bumps all over it. There are little red dots in our diagram. Those are ribosomes. That's where we're going to make proteins, because ribosomes make proteins. The smooth ER don't have the little bumps on them. Oops, sorry, pointing the wrong place. Smooth ER is over here. And those, since they don't have ribosomes, that's where we make our fats, the lipids. So in our factory, that's the site for product building, whatever our factory is building. Then let's talk about more specifics, the ribosomes. Those are those little red dots on here. So we'll call those the factory floor. They're the site where actual protein building happens, protein synthesis, and it produces all the proteins. We produce all the proteins we need for our cell on those little teeny red dots. So whatever you're making in your factory, that's where your product's being created. The Golgi apparatus, it modifies, sorts, and packages. So in a factory, that would be packing and shipping probably. Maybe it's labeling, packing, and shipping. And so this Golgi apparatus, always my favorite organelle, I have to admit, gets this transport vesicle of proteins, and say we want to change it a little bit. We add something to it. It's using, this is the base protein, and it might add a few functional groups to change it into something else, and then packages it and ships it out. Lysosomes are these wonderful things that they're the only ones that don't create something in the whole cell, the organelles. Lysosomes digest. So if we have macromolecules, we've saved energy in, remember from last module, we have a starch molecule, say if your plant had a starch molecule saved, this guy, that's going to be the one that breaks it down so you can access that energy and grow in the spring, right? So lysosomes are going to do that. They're also going to break down, if we have old cell parts that aren't working very well anymore, got too old, we need to replace them, it breaks those down into raw ingredients so we can use them again. Lysosomes, really, really important. I already said this. They're actually producing nothing, and they're vesicles produced by the Golgi apparatus that serve to digest molecules or portions of the cell. So my factory analogy, I was thinking those are maintenance. They go and clean things up, fix things, rebuild, break things down. So got the gist of that. And so how does a protein go through there? The correct sequence is it's produced in the rough ER on the ribosomes, it's shipped to the Golgi apparatus in a vesicle, and then it's maybe modified, put in another vesicle, and then moved to the plasma membrane, where it kind of just merges when it does it. They don't have a picture here, but it just comes to the plasma membrane, and the membrane of that vesicle merges with it, and then it opens to the outside. It's kind of cool. So from here to here to here, and then to the outside probably. Vacuoles, how are they different from vesicles? Well, vesicles are smaller. They're both membranous sacs. Vacuoles tend to be larger, and the one that you'll hear most about are the central vacuole of a plant. This is what makes your plant turgid, stand up and not be wilted. And so it keeps extra water in there, as well as other nutrients and minerals storage. So basically vacuoles are storage. So factory analogy, storage of building materials. And plants, while we're talking about them, have a cell wall. This is this green around the outside in this image. It is thick, strong cellulose. It gives the plant a shape. Animals do not have cell walls, only plants do. And then in the prokaryotes, bacteria do. So in our analogy, this cell wall would be like the exterior structure of the factory. And if you see this plasma membrane here, that's that little kind of yellow-orange lining inside here. That's what animals only have. They don't have a cell wall with it, but both plants and animals have that plasma or cell membrane, which we'll talk about in a little bit. All right, so let's talk about mitochondria. I think you've been hearing about that your whole life probably. The powerhouse of the cell, the currency of the cell. It's what makes the energy that makes everything go. And so actually it's releasing energy through cellular respiration. So this is the mitochondria right here, and it produces ATP. It breaks down your food that you've eaten and releases the energy from it so you can do whatever it is you need to do. Our factory analogy sometimes is it's the money, because it's what runs everything here. Plants and animals both have mitochondria. They both have to break down that energy and use it. But plants are those magic ones we couldn't live without that capture solar energy through photosynthesis and converts it into a chemical molecule with energy. Otherwise known as glucose is a good example. And so the guys that do this are right here, chloroplasts. See those green guys? In the factory analogy, that you might think of that as the contract for work that sets up payments and collecting payments, because it's not exactly the money yet. It's not what's running it, but it's what's getting us the money. It's the last step before it. So where would we find a whole bunch of mitochondria? Recognize that? You betcha. That is a sperm. That little guy has a long way to swim. Big time. And so this whole flagellum back here, flagella, is full of mitochondria. So it can release ATP like crazy and keep on going till it gets to its final destination. And then if you like poultry, if you like eating chicken, the thighs are called dark meat. They're darker in color. That's simply because they have more mitochondria, because that's a large muscle of the body for those chickens. Kind of interesting, I always thought. So folding in here, inside the mitochondria, has a whole bunch of folding. Once again, more surface area. We just want more surface area, and then we can create more ATP, releasing it. So the cytoskeleton is this whole interconnection of the cell. It maintains the shape of the cell some. It assists with movements of cell parts and organelles. The organelles actually sometimes move. Remember those vesicles moving? They move along the cytoskeleton. So it's this network of interconnected, these protein filaments and tubules. So if you think of it like a major highway system inside that cell, you actually have it right. In our factory analogy, we'll think of it as a communication system within the factory. It could be even the halls and corridors that connect each section. So here's kind of a picture of it. It's this reddish stuff. And so we have these things, these fibers that connect all the way through. So let's say we had something produced on this endoplasmic reticulum, on that ribosome, put in a vesicle, boom, and it's kind of bumped along that until it goes over wherever the Golgi apparatus is. So, highway system. What organizes it, makes that superhighway? That's a centrosome. A centrosome is kind of a weird thing. We actually don't know completely about it. It kind of surprises us. I think we think, oh, maybe it does this, but it does manage the building of all those fibers inside there, of that superhighway system. And when we talk about mitosis, it's also what helps organize and make the centrioles and all this whole business that creates, divides the DNA. So let's think of that as processing headquarters, that it organizes the movement of material to factory floor and products away from the factory floor. So intracellular proteins, so these are those microtubules I'm talking about, and they're made of actin, myosin, they're microtubules, and keratin. Those are the main ones. And it's actually ATP works directly on it to bounce that organelle along. It's a chemical reaction that makes it move. Let's talk about extracellular matrix just really briefly in animal cells. This is the stuff that's outside the cells, and it's usually secreted by the cells that it's close to, and it kind of helps hold all things together, the different cells. It's communication, it provides consistencies from cartilage to bone. It helps cells just really exist. And in addition to that, we have some junctions. So there's an adhesive junction. These are all proteins. Remember, proteins, we're going to become those ribosomes, really important. So these are ones that hold cells together. Like if your heart, it has to pump and stretch, your stomach, your bladder, it's going to hold things nice and tight, but not watertight yet. These are just adhesive junctions. They're kind of a different one. Then you have tight junctions. These are these zipper-like things. You can even see they look really kind of like this picture. And it serves as a waterproof barrier. So we also find out their adjacent plasma membranes are joined. So that's kind of a big deal, having all these plasma membranes hooked up. Then we also have plasmodesmata, and those are for plants. And it allows, here's some actual pictures of them and slides I thought was pretty darn cool. It allows plant cells to communicate between each other and connect the cytoplasm of one with the other. I mean, these are living organisms we're talking about, and so a cell really usually does not exist alone. It has to work with a bunch of cells coming together, make a tissue. A bunch of tissues come together, make an organ. A bunch of organs come together, make an organ system. A bunch of organs working together, organ systems working together, make an organism. So let's talk gap junction. We need to communicate. And so they actually even look sort of like that. See the real slide of it? It's crazy. And so those are just ways that it's actually a chemical response where how they communicate with each other. I want to throw in a little bit about cilia too. I think that you're familiar. You might know that they're in your nose, you're in your windpipe, and these little finger-like projections here, they just keep the passageway clear of debris and keep things out. And if something gets in there, they all kind of work together to move it towards the exit again. So what organelles have membranes in summary, kind of? Main ones we've talked about, nucleus, chloroplast, mitochondria, and lysosomes, and there's probably more. But the one I wanted to bring to your attention is ribosomes don't have a membrane. It's kind of an odd thing, but they do not. All right, what do all cells have? Prokaryotes and eukaryotes, they've got a cytoplasm. That's the first thing I started with you with. They've got a plasma membrane, also called a cell membrane. They have DNA, and they have ribosomes. And here's a few pictures so you can practice identifying things. Now let's talk a few minutes about the plasma membrane. Both prokaryotes and eukaryotes have this. Let's see how it works. We have a polar head, and then we've got these nonpolar tails. The whole thing together is called a phospho, because this is a phosphate, lipid, because these are lipids, these are fats. So here's what this whole membrane looks like. It kind of looks like an Oreo cookie, and there's 2 rows of these molecules on there, and the phosphate heads point to the outside of the cell and inside, and then the hydrophobic area, right here, the tails, face each other. So what we got here, something is going to have to go through hydrophilic, hydrophobic, then hydrophilic. It works out to be an awesome barrier that not many things can get through. Oh, I was telling you Oreo cookie. The phosphate heads are the cookie, and the creamy part is the hydrophilic tails. So, molecule passes through it by going, and it's kind of cool that these polar areas on the inside and outside, so it'll track the molecules that they need. Then it won't let them through. So it's this wonderful gate. And then these proteins we'll talk about in a minute are the main things that move, this one in particular, that move those molecules through. And so it has to go through without that protein through that hydrophilic, hydrophobic, hydrophilic again. Okay, picking up our plasma membrane or cell membrane, here's those double layer, the phosphate heads, inside and outside, nonpolar hydrophobic center there. Oxygen, carbon dioxide, and other nonpolar small molecules can go through here. Big things like glucose, ions that are highly charged can't go through. And water really doesn't. We actually have these special aquaporins that move water back and forth for us. So since it blocks everything that except for those few things, we have to have a way to get what we need in and out. So we have these things called channel protein and carrier proteins, and they bridge across the whole membrane. And so a channel one just goes with the flow. It needs this kind in here, it's specific for that molecule, but if it's more concentrated out here, it'll move them across, so there's more inside. No energy is used. But a carrier protein can go against that flow. And so it needs to take some energy to do it, and it captures a specific type of molecule, just like this one does, but it takes energy to transport it in and release it. It takes ATP, this guy does. Because this is the way we're going to get what we need across that barrier, because it's an awesome barrier. Here's another picture of one of those channel proteins right here. Now, glycoproteins are pretty darn cool. That's this guy right here. It's this protein that's in here with a carbon chain. Glyco is sugar, so the sugar chain sticking on the top. Guess what? That's what is like the fingerprint of your cell. It says that's who you are, what you are. And so if you have an organ transplant, this is the guy that we got to worry about, because this is not recognizing this is what's going to make an organ rejection happen. So you have to take special medication that stops that recognition. And so this whole thing here is all about you're in the right place at the right time, recognition of what it is. And receptor proteins do something else. They're kind of darn cool too. If you think about different hormones that have biofeedback systems in our body, such as insulin, that then moderates our blood sugar and lets us know when to release more, when to, not to release as much. That's from these receptor proteins that are embedded in the membrane again, the cell membrane. So they have a certain ligand, which is a messenger that comes and fits in here, and that causes another reaction down here. And then if something needs to be created, like if it's saying, oh, we need some more whatever, the hormone causes it to be released, then your cell goes ahead and makes it. So those are what receptor proteins do. They're kind of amazing things. And I'm done.